like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lodin School of Medicine, our Healthy Longevity webinars. Thank you for being us and with us and joining us. Today we have a special guest. As always, it is Professor Bjoern Schumacher. He is with us tonight and we will learn lots about C. elegans. It's a very tiny little worm and about what we can learn from that worm with regards to aging. You might have lots of questions, so please use the Q&A function to either have the questions or leave comments. It's highly appreciated. First, we will start to hear from Indrik Vijaya. He is the research assistant at our Center for Health Longevity, and he is quite interested in aging biomarkers, and he will present an aging clock based on inflammatory markers, and it's called IH. And he will show how that is associated with relevant outcomes. Indrik, the floor is yours.
Thank you for the kind introduction. As mentioned, I'm Indrik Vijaya, a research assistant in the Center for Healthy Longevity. And today, I'll be sharing with you a fascinating application of AI on aging research done by investigators from the Stanford University and Buck Institute. The team developed a new AI-based biological clock called the Inflammatory Clock of Aging, IH, that can predict age-related inflammatory diseases and measure the overall health system of the immune system with greater accuracy than the number of candles on your birthday cake. As we grow older, bad inflammation, which is closely linked to our immunity, begins to kick in, which then often leads to organ damage and causes a wide range of diseases, including cancer, heart attack, strokes, and neurodegeneration. While this has been widely known, immune metrics that are capable of identifying the most at-risk individuals are lacking. Thus, this research is significant as it provides a potential for high-quality longevity and validated biomarkers, allowing us to predict how soon you'll become frail. A substantial merit of the study published was the robustness of the evaluations performed on 1,001 individuals aged 8 to 96 years old, the multivariate data collection, data harmonization, and subsequent data analysis. Extended clinical report forms were also collected from 902 individuals. Then deep immune profiling includes cell enumeration, proteomics, functional phenomics, and gene expression data. The group also measured circulating immune proteins, cellular responses to cytokine stimulations, signaling pathways, and levels of CD8 plus naive T cells, which is a hallmark of immunosenescence. Subsequently, they used a guided autoencoder method, a powerful AI algorithm to correlate this combined information with multiple readouts of aging. This method is a type of deep neural network that utilizes nonlinear equations and effectively eliminates noise and redundancy in data, yet retaining the most relevant biological information from circulating immune protein data. Using this method, the derived IH accurately predicts chronological age in the population. Then 10 age-related disease items were selected to characterize the clinical significance of IH. The items analyzed included different diseases and physiological systems such as cancer, cardiovascular, respiratory, reproductive, and psychiatric. After adjusting for covariates such as age, sex, body mass index, and other biological factors, IH was significantly correlated with multimorbidity in the older population analyzed. This highlights the key role of IH in the accumulation of physiological damage during aging. They also found that the total amount of certain T cells, B cells, and white blood cells that play a role in immune response demonstrated a similar correlation. The research then also observes that a set of about 50 immune signaling proteins called cytokines were the strongest predictors of IH. And notably, IH clock picked the chemokine protein CXCL9 as being the most significant contributor to a person's score. The investigators then compared CXCL9 levels between different age groups and found significant increases in this protein with age, starting at the age of 60 years old. It is known that CXCL9 elevated levels have been associated with autoimmune response, host immune response, and multiple other inflammatory conditions, highlighting its status as a valuable biomarker of disease in general. And importantly, CXCL9 and several of these markers have been linked to cardiovascular aging and are known to be secreted by endothelial cells. Nonetheless, a lead study suggests that reducing CXCL9 levels will restore youthful endothelial cell function, indicating that CXCL9 directly contributes to those cells' dysfunction, and that inhibiting it could prove effective in reducing the risk of cardiovascular diseases. To sum up my presentation, we have a few key points that we have learned from this presentation. Firstly, by applying AI methods to immune monitoring of human blood, we can have an inflammatory aging clock, which can be used as a companion diagnostic to inform physicians about a patient's inflammatory burden and overall health status, especially those with chronic diseases. Secondly, IH assists in identifying the risk of healthy older adults with no clinical or laboratory evidence of CVD for early cardiovascular aging. And lastly, CXCL9 has a high potential for therapeutic target that can prevent age-related deterioration of the vascular system and other physiological systems as well. That's the end of my presentation. Hope you have enjoyed this presentation. Thank you for your kind attention.
Thank you. Oops. Thank you, Indrek. Wonderful. I think the future is really in these kind of clocks, and hopefully we will implement it into clinical practice soon. But now over to Professor Bjoern Schumacher, our guest. He is, I would say, famous for his studies on how organisms really adapt to the accumulation of DNA damage with aging. He has lots of affiliations. He is the director of the Institute for Genome Stability in Aging and Diseases at the German University of Cologne. And he received lots of prizes, under which the Eva Louise Köhler Research Award. And he also received an ERC grant, that's the European Research Council grant. And he was the coordinator of a very big consortium, which was the FP7 Marie Curie training network, where the network really looked at chronic DNA damage in aging, which is, I think, very important. He is the president, or he was the president, actually, of the German Society for Aging Research. And currently, he is the vice president of the German Society for DNA Repair. And he is also the co-director of the Minerva Center of the biological mechanisms of healthy aging at the Balaam University in Israel. And I can tell you, I'm following him on Twitter. He really likes to Twitter. So, and his tweets are very funny. So you might consider in following him on social media. So Björn, it's wonderful to have you on the show, and wonderful to learn from C.elegans from you. So the floor is yours.
Thank you so much, Andrea, for the very kind introduction and for the invitation here to speak today at this very highly international worldwide forum that the National University of Singapore and its new recent activities in really becoming a hub, a major hub for aging research has established here. And I also like to thank Max and Brian for together with Andrea to set this up and host me here today. So I would really like to talk to you about what we learned about genome maintenance mechanisms in aging. And as Andrea already mentioned, our favorite model here, C.elegans. In this worldwide forum, I think it's needless to say that we are living in a worldwide demographic change. The populations are getting older, not only in Europe, not only in North America and the wealthy nations of Asia, but really worldwide. So we share this issue. Now, aging, having a long life expectancy is certainly one of the major achievements of modern civilization. However, most people with age will become chronically sick. And in fact, the majority of the elderly suffer from multimorbidity. And these are really the challenges that we need to address. Multimorbidity, multiple chronic diseases occurring at the same time, is exponentially increasing with age. This ranges from dementia, with Alzheimer's the most prevalent one. Cardiovascular disease remain the number one killers. Chronic kidney disease is a major clinical issue. Type 2 diabetes is on the rise. Cancer, most cancer types are aging related. Age is the number one risk factor for cancer, osteoporosis, frailty, and so on. It affects the majority of the elderly. Already nowadays, over half of all healthcare costs are spent on the elderly. And by 2050, we will have 2 billion people living above the age of 60. The traditional approach for tackling age-related disease has been really specialized treatments. Treatment for cancer now through personalized medicine. We are in the era of precision medicine, specialized cancer research, trying to understand the individual biology of the cancer to improve treatment. This is true for every individual treatment of that. Now, if you bring that together with the multimorbidity, this approach is not going to work in the future. Instead, the modern aging research has shifted the paradigm here, because these diseases are not only associated with aging, but we actually postulate that aging is in fact the root cause for these distinct symptoms, I would call them, of aging. And so when we devise treatments that target the aging process instead of individual diseases, we will be able to protect against age-related diseases, lower the risk factor, extend healthy lifespan. And this clearly will be the future of medicine. This, however, requires that we really understand the mechanisms of aging. And if we want to understand anything in biology, we have to consider the process in the context of the evolutionary history that has shaped it. And here a key realization came in the late 19th century by August Weismann, a zoologist who back then was one of the top 3 evolutionary biologists after Darwin and Wallace. And he conceptualized the concept of inheritance. And he proposed, which we all know nowadays is that it's true, he proposed that germ cells and only germ cells perpetuate the genetic information throughout the generations. And we as humans are almost the 7,000th generation now of modern Homo sapiens. And in fact, this has been one line of germ cells that are essentially living on indefinitely in us. Essentially, Weismann realized immediately that because germ cells live on indefinitely, indefinite maintenance, immortality of the soma would offer no benefit in terms of the fitness of a species. Therefore, upon installation of the next generation, which in humans takes about 3-4 decades, then the next generation is fully functional, can take over. There's no longer any maintenance required. The organisms can age, decay, and die. And that's essentially the bottom line why we age. Now, Tom Kirkwood proposed the disposable soma theory of aging, which really neatly conceptualizes the concepts about what the issue with the soma is, because the soma needs to be maintained only to allow successful reproduction upon which it can be disposed of. There are now three questions arising from this concept, and these are the questions that are really at the heart of the interest in our lab. The first one is, which are these processes that determine somatic maintenance and thus control aging? The second one is, how is the somatic maintenance adapted to the requirements of the germline? Just imagine what would happen if a human were to age within 1 decade of his life. There would be no reproduction, species would be terminated. And in fact, there are humans that exactly suffer from that. And finally, of course, the ultimate questions: how do these germ cells manage to live on forever? Now, why is DNA damage so important here? DNA damage is constantly occurring. There are tens of thousands of lesions occurring every single day in every single cell of your body. So it's constantly occurring. It can come from the outside. It can come from very normal metabolism. And there's a myriad of different types of DNA lesions. And these have distinct consequences. We know for a very long time, for many decades, we know now that DNA damage can lead to genetic aberrations, most prominently mutations that alter the gene sequence. And when tumor suppressor or oncogenes are affected, this will lead to cancer. Cancer is caused by DNA damage. We know this for a very long time. But DNA damage can also block DNA metabolism. It can block transcription. It can block replication. And this has profound consequences on the cells. The cell fates are affected by that. Cells induce cell death, apoptosis. I will talk about that. Cells also undergo senescence. It's a bona fide DNA damage response. Stem cell compartments get deprived. Some cell types undergo polyploidization. And this is exactly what is happening in aging. This functionality of cells decline, tissues become atrophic, and inflammation becomes systemic. So DNA damage not only causes cancer, but also drives forward the aging process. And in fact, with increasing evidence has linked DNA damage to every single one of those so-called hallmarks of aging. It is the cause for genome instability. Shortened telomeres, dysfunctional telomeres are essentially a specialized form of DNA damage. The epigenetic code is affected by DNA damage and is altered upon DNA repair events. There's proteostatic stress observed in the DNA damage response, and also mutations can lead to protein misfolding, and mitochondria become dysfunctional in the response to chronic DNA damage. The cellular consequences, stem cell exhaustion and cellular senescence, are consequences of a DNA damage response, and this has systemic consequences. Signaling mechanisms are disturbed, such as endocrine shifts that occur in mammals. Inflammation becomes chronic in the DNA when DNA damage is not removed, and nutrient signaling is dysregulated. Now I have some problems here. Now it's there. There seem to be some issues here with the. So let's get back to our three questions. I will briefly give you examples for the first two, and then discuss in more detail how we approach germline genome integrity. So to answer the question, which processes determine somatic maintenance and will ultimately control aging, I just showed you an example here of DAF16. DAF16 is the FOXO transcription factor that, when it's active, promotes longevity. It was the first factor ever found to extend longevity in a highly conserved manner. Normally, DAF16 here marked with GFP in a worm is cytosolic, doesn't do anything, is inactive. But when we induce DNA damage, we use UV here as a paradigm DNA damage, because this type is extremely well characterized as the lesions that it induces. We find that now DAF16 is activated. It responds to DNA damage. Now we undertook a phosphoproteomic approach to get a comprehensive view of what is happening in the DNA damage response. And here are alterations induced in red, repressed in blue, that are either at the phosphorylation or on the proteome level altered in the DNA damage response of the animal. And what you can appreciate here is that all these responses evolve around two major regulatory pathways of longevity. Insulin-like signaling at the very core of it, and the TOR signaling mechanisms that are responding to DNA damage, and they are linking now to physiological alterations. And these are the dampening of proteostasis and metabolism that are repressed in this paradigm here of unrepaired DNA damage, and an induction of chromatin remodeling and of autophagy. And what are now the consequences of longevity assurance mechanisms that respond to DNA damage? This we addressed here, and let me take you through this slide. It's just an example to demonstrate this. Let's start with this one here. So we have your wild-type animals that are treated with UV, and we here with antibodies can detect the cyclic return pyrimidine dimer, the 6,4 photoproducts, the lesion types that are formed in the chromatin here, and they are removed within 24 hours. Now we have a repair mutant. This mutation here in XBA is a repair defect that also exists in humans. And this now prevents completely the removal of DNA damage. Now we add to that a mutation in DAF2. DAF2 is the insulin-like growth factor 1 receptor, and a mutation in DAF2 extends lifespan in C. elegans and as well as in mammals. And here, of course, the lesions are not repaired either because there's a repair defect. Now we look in adult worms at the functionality of the tissue, pharyngeal pumping, motility, and we find that in the repair-deficient animals, upon very, very low doses of UV, we see a steep decline in these parameters of functionality of the tissues. This is completely prevented by the mutation in DAF2. What does this mutation do? It leads now to constitutive activity of DAF16 FOXO. And this DAF16 is a critical factor. It's now constitutively active, and it completely prevents the loss of tissue functionality. Likewise, post-UV survival is greatly extended. So despite no removal of the DNA lesions, this led us to postulate that there are essentially two mechanisms of longevity. There's on the one side, DNA repair. So when you imagine that now DNA damage occurs all the time, it will gradually increase over the aging process, and at some point it will surpass a threshold where tissues can no longer function. DNA repair is absolutely essential to slow down the accumulation of DNA damage. We can have in humans a single DNA repair defect, and damage accumulates within the first decade of life, and by 10 years of age, these patients look like they are 100 years old because they suffer from all the age-related diseases we get in normal aging. Longevity genes, such as the insulin signaling, the DAF16 transcription factor, do something profoundly different. Instead of slowing down the accumulation of damage, they raise the threshold when the damage becomes detrimental for the survival of an animal. So they raise the threshold of until when DNA damage can be tolerated. It's a DNA damage tolerance program, and that allows now plasticity of aging. It allows these animals to withstand the damage accumulation for longer. Now let's just have a glimpse at the second question. How is the somatic maintenance adapted to the requirements of the germline? And here, of course, C.elegans is a fabulous organism to answer this very, very complex type of questions. And this was done by Maria Ermoleva, who now heads her own research group at the Fritz Lippmann Institute in Jena. And she asked the question here, what would happen here in a schematic representation, one half of the germline of C.elegans, how would the soma respond when there was now damage in the germline? And I just give you the summary of this, where we found that DNA damage in germ cells induces an innate immune gene response that becomes systemic throughout the entire animal. It activates the ubiquitin proteasome in the somatic tissues, enhances proteostasis, which leads to systemic stress resistance. And this augmented somatic endurance now allows time for the animal to repair the damage in the germ cells. It extends reproductive lifespan to allow the animals to later in life, when normally reproduction has ceased already, to now produce offspring with repaired genomes. Is this applicable in humans? Now you can think about some translational aspects, how to get the treatment out of that. So first of all, does the germline play a role in human aging? And the answer is absolutely it does. Here is a study that comes from the historic records from the Korean court, the royal court, where there were eunuchs that had actually a relatively normal lifestyle, an aristocratic lifespan, and because it was known how long these eunuchs lived in historical times, their life expectancy could be now compared to those of male members of aristocratic families. And indeed, there was nearly a 20-year lifespan extension. The interesting thing that what we learn from C.elegans, it's not about fertility itself. It's about signals between the germline and the soma. And I think it will be very important to figure these out in mammals, where, of course, they are more complex. Now I want to just spend the remainder of the talk on what we learned about germ cells. How can germ cells be immortal? And it is clear that the genome stability in germ cells is a prerequisite for fertility, germline immortality, and eventually for species maintenance. Now, C.elegans is a fabulous model to study this because it has this complete distinction between the post-mitotic soma and this highly dynamic germline where mitotically expanding germ stem cells then transit into meiosis, where they form oocytes or self-fertilize to form embryos. And the DNA damage response occurs in two sections here. The mitotic stem cells, they undergo cell cycle arrest, while specifically the meiotic packaging cells, is the phase in meiosis where recombination happens, undergo apoptosis that you can now see in live animals in the germlines upon DNA damage. And this allowed us now to investigate how somatic mechanisms, how the soma that also surveys the environment for stress factors and so on, how that might impact the apoptotic control that controls the genome integrity of germ cells. And this project was done by a very talented grad student, Najma Sultan Mohammadi, who was joined by an exceptional postdoc, Xiao Wang. And to visualize this, we look here now at these germ cells, at this band where the packaging cells are, and after ionizing radiation that induces double-strand breaks, we see these apoptotic corpses. When, however, we look here at a mutant of the P38 stress signaling pathway that's called PMK1, that's the P38 ortholog, we find a very strong defect in forming apoptotic corpses in the germ cells. Now, in fact, the PMK1 is activated. Here, phosphorylation increases after ionizing radiation. It's very specific to PMK1. It's absent in the mutant. And we find that when we score here now the apoptotic corpses in the germ cells that greatly increase after ionizing radiation, this is completely defective in PMK1. The downstream transcription factor of PMK1 ATF7 is likewise defective, and in unrelated MAP kinase JNK1 is expendable for that. Now, when we now look at the in situ the activation of PMK, we identified that this is actually not at all, it's not even present in the germ cells. The germline doesn't have any PMK1. Instead, when we look here at the activation, phosphorylated PMK1, we find it induced upon DNA damage in the intestine, but not in the germline. So the germline is completely devoid of that. And we can quantify that here. There's a strong induction, which is specific to PMK1. Now, when we now rescue this mutant of PMK1 that is defective here in apoptosis in the germ cells, and we re-express PMK1 only in the intestine by an intestine-specific promoter, the wild-type copy we restore germ cell apoptosis. We can also do a completely independent experiment here by using RNAi deficiency strains that are completely RNAi defective, but we restore their ability to do RNAi now cell type specifically, either in the germline or in the intestine. And we see here when we RNAi PMK1, we have a drop in the apoptosis, not as strong as in the mutant because it's only RNAi. But when we RNAi it in the germline, there's no effect. But when we RNAi it, when we knock it down in the intestine, then germ cell death is reduced. Now, we see this also upon endogenous meiotic recombination failures. This is a mutant that has recombination failures, repair defects during meiosis, endogenous, and here also this is strongly reduced by a mutation in PMK1 and restored when we have intestinal PMK1 expression. Now, if we indeed find that a downstream target of PMK1, which is a previously uncharacterized gene that we call CISM1, and this target is strongly induced after ionizing radiation and completely dependent on P38 PMK1. And we find that a mutation in this target of PMK1 signaling that we call systemic stress response mediator, that this mutation has a defect in apoptosis, and we can restore apoptosis and even hyperactivate it by now bringing back a wild-type copy of endogenous CISM1. Now, when we restrict the expression of CISM1 in the mutant, so we have a mutant of CISM1 that is defective in DNA damage-induced germ cell apoptosis, we can restore this by either constitutively expressing CISM1 in the intestine, where PMK1 is active, or use a germline specific promoter where constitutive expression of CISM1 in the germline also reinstates apoptosis. Now, CISM1 is indeed acting downstream of PMK1 because we can suppress the apoptosis defect by now re-expressing CISM1 in the intestine. Now, just bear with me for 1 second, where here we produce now a range of CRISPR-Cas knock-ins in the endogenous locus of the CISM1 gene. So a mutation of CISM1, a deletion, leads to the defect of apoptosis. We can tag CISM1 by knocking in the tag. Then we can swap the promoters to an intestinal promoter or a germline promoter. And then we can, in those strains, eliminate the signal sequence that is usually associated with the secretory pathway. And indeed, what we find is that all these different promoter-driven CISM1 are normal in inducing apoptosis in germ cells, unless the intestine-specific CISM1 lacks a signal sequence, then there's a defect in germ cell apoptosis. And indeed, what we find is that the wild type, the normal CISM1, is upon ionizing radiation associated with the secretory pathway. Here, we look at one intestinal cell where it co-localizes with the Golgi marker. And indeed, what we see is that CISM1 is induced after ionizing radiation in the intestine, and then it translocates into the germ cells. And this is defective when it's intestine specific now produced, but has a mutation in the signal sequence. Then it's only present in the intestine, but fails to be transported to the germ cells. Now, we then ask whether there's any stress factor that would be important for PMK1 to sense it, an environmental stress factor that is sensed by PMK-P38 stress signaling. And here, a transient heat stress of only 10 minutes prior to inducing DNA damage by ionizing radiation, these 10 minutes of elevated temperature is sufficient to induce apoptosis in germ cells and greatly exacerbates the apoptotic response to ionizing radiation, to double-strand breaks. This is completely defective when PMK1 is absent, and it's reinstated when we re-express PMK1 only in the intestine. The same is true here for CISM1, where we see in the CISM1 mutant a strong reduction in the stress-induced apoptosis, and this can be reinstated by intestinal expression of CISM1, and it's sufficient to restrict CISM1 expression to the intestine for this. Now, what is the consequence of genome instability, of DNA damage in germ cells? The consequence is aneuploidy. And this is something that in humans, for example, age-dependently increases, particularly associated with female reproductive aging, where aneuploidy in humans, this is trisomy, increases with age. And in C.elegans, aneuploidy can be measured by a so-called Him phenotype, where one of the X chromosomes is lost through chromosome non-disjunction, and in the progeny, you see a high incidence of males. Usually there are no males, nearly no males in the progeny. After a transient heat stress, we see some male production, some aneuploidy in wild type. This is great. This is significantly elevated when PMK1, when the stress sensing pathway in the intestine is defective, and we can revert the occurrence of aneuploidy by now reinstating PMK1 only in the intestine. The same occurs after ionizing radiation, DNA damage of the germ cells. There's aneuploidy occurring in wild-type animals, significantly elevated in PMK1 mutants, and we can completely revert the aneuploidy by intestinal expression of PMK1. So the conclusion of that is that the PMK1-P38 pathway in the intestine regulates DNA damage-induced apoptosis in germ cells. PMK1 responds to environmental stress and germ cell DNA damage to promote the apoptotic response and the quality control during oogenesis. CISM1, the effector of P38, is secreted from the intestine into the germline to regulate apoptosis. The intestinal PMK1 controls stress-induced aneuploidy, and this indicates that somatic stress signaling regulates the control of heritable aneuploidy. And just for some take-home messages, what I showed you today is that somatic DNA damage tolerance program is mediated by a DAF16-centered longevity network. Somatic maintenance mechanisms respond to germline DNA damage, but reversely, the soma regulates the stability of heritable germline genomes, and this essentially challenges the Weismann barrier that I told you at the very beginning about, that germ cells and only germ cells determine genetic inheritance, but instead somatic stress signaling greatly influences the inheritance of chromosomes and of the genetic information. I acknowledge the people who did the work, and I thank very much the audience today. I apologize for the few hiccups. It's a brand new computer. Technology should usually enhance our lives, but all too often it doesn't. So with that, I close and I get back into the Zoom.
Nice. May I ask you first, because C. elegans seems to be a wonderful model organism because it lives quite short compared to the human lifespan. And you started with these wonderful slides actually showing that with the aging process, we accumulate so many age-related diseases. What kind of diseases does C. elegans accumulate? I know it's being used for sarcopenia research, but tell us a little bit before we go into the DNA damage, what kind of phenotypes you see in C. elegans.
Yeah, so sarcopenia is very obvious because the muscle degenerate very, very similar to human muscle degeneration. It shows the lipofuscin and all these things. But what I find most strikingly is the neurodegeneration, because in C. elegans, a third of the somatic cells are neurons. So in relation, they have much more neurons than we have. But what we see is really striking neurodegeneration with the very same processes of progressive neurodegeneration that we see in humans. And we can follow that in life in these animals, over their aging trajectories. Different neurons are also differently susceptible. And we recently actually found that very similar to the Prodroid patients that have a DNA repair defect, the same mutation in a worm leads to strongly exacerbated neurodegeneration. It happens already in early adulthood, just like in the human patients, that neurons progressively degenerate. So they are first all normally built, but during early adulthood, they already show axonal degeneration. And so that, I think, is an aspect that can be that is very, very similar in C. elegans. And in my view, probably for neurodegeneration, it's one of the premier model organisms to shed more light on that. Okay,
so we should stop the research in mice, in all the mice models looking at dementia, but focusing a little bit more on C. elegans? Okay.
It's actually much more profound in C. elegans. It's very, very similar to the human situation. And I think it's a very, very important expansion. I mean, I wouldn't say abandon it, but I think we should really widen our horizon, particularly for really getting some key fundamental insights into it. I think it's a super relevant model for that.
Yeah. So let's switch to DNA damage. And I have this wonderful position that I can now really ask you, how important is DNA damage compared to all the other hallmarks of aging? And look, you already showed a slide where you really focused in that slide in the center, it was DNA damage. But I can tell you, if you would ask somebody else, focusing a little bit more, for example, on methylation or epigenetic alterations, they would also say this is in the center. So tell me a little bit, how much is really due to DNA damage compared to all the other hallmarks of aging when we are talking about C. elegans lifespan or the human lifespan?
Yes, even if it challenges some concepts and some ideas, I think it's important about really setting apart the arguments. What is so special about the genome? First of all, DNA damage always occurs. There is no such DNA molecule in a human cell that doesn't have damage, and it requires constant repair. That's the first thing. It has profound consequences of anything in the cell. So it turns out the DNA damage response really affects every physiological process. That's something that that has become very evident. Then you have to go to the human genetics. And when you increasingly look at the Prodroid syndromes, so where aging is accelerated, sometimes in a segmental fashion, so where there are some specific cell types age prematurely. In some, like cocaine syndrome, for example, it's really widespread. So they have premature aging in about every tissue you look at. They die of neurodegeneration and arteriosclerosis within the age of average 12. And all they have is one mutation in a DNA repair gene. Everything else is not altered. And that, I think, is a very striking argument for that. And you find this in every Prodroid syndrome that you look at. You find genome instability. Now, also, if you look at neurodegenerative diseases and dementia, you find DNA damage. It is just so clear that it's such a fundamental level that it relates to all these other hallmarks, and it is sufficient to promote the entire aging process, while other things might promote aging in a specific cell type. But here it's really every tissue, every organ ages.
Okay, so it seems to be very central. So that's good. But tell me then a little bit, because we were talking about slowing of the you have, on the one hand side, you have the damage, and on the other hand side, you have the repair. And then I think translated to that, you have DNA repair genes, you have longevity genes. So what is the best balance for longevity in different model organisms? So how much of what should you have?
Yeah, so that's a very interesting question. So I think the major distinction is between germ cells and somatic cells. I think that's a major thing that sets it apart. That germ cells have about 2 orders of magnitude lower mutation rates than somatic cells. And that is because there are 2 reasons for that. The number one reason is their DNA repair systems are functioning much better. They have the same, of course, as a soma, but they just make more of that. They repair more accurately. And the other one is selection. That, for example, during oogenesis in humans, there's a very, very strong selection for, we know that oocytes are eliminated upon just occurring a few double-strand breaks. Huge problem in cancer therapy, for example, particularly for young women. So there's a very, very, very low threshold of tolerance to DNA damage. And somatic cells, of course, they adapt. They are very different. I mean, stem cells, for example, have different genome maintenance mechanisms than differentiated cells. Recently, in the past 1 or 2 years, we realized how much DNA damage physiologically occurs in neurons. It's completely, it would have been unimaginable just a few years ago that during transcription, for example, you constantly induce DNA damage on a very physiological level. I think these balances are very important. What type of damage leads then to apoptosis? If every DNA damage led to apoptosis, you'd have neurodegeneration before you turn 1 year old, right? So essentially, this balance between threshold setting, the DNA damage response, and repair is something profoundly important. And what we are aiming at is really in the future to be able to augment DNA repair. I think that will be, it's a very, very complex process because there are so many different DNA repair mechanisms, different types. But I think it's extraordinarily important if we want to maintain health, also not only of stem cell compartments, but also of differentiated neurons. We will need to augment DNA repair.
Yeah. So before asking Sarcopenia, who is our research assistant at the Center for Health and Longevity, what the audience would like to ask, one more question from my side. If DNA damage is so bad, how much is avoidable through external stressors, and how much is, you call it even physiological, but what is the ratio? How much is in our own hands to avoid UV, etc., or any idea?
So that's a very, very important question. This will be different in different cell types. So you mentioned UV already. I mean, we know that sunbathing is not only the number one risk for carcinogenesis, but also for skin aging. So avoiding UV light is very important, but this goes, of course, beyond that, because you also have a lot of dietary components that induce DNA damage. We know nitrosamines, so avoid burnt like barbecue meat, right? Things like that. We know that they induce nitrosamines that interact with DNA directly. Smoking is so clear how adverse the health effects of smoking are, because there is DNA damage. So there's a big component of avoidance that I think we need to take more seriously, because also in the diet, it's very complex to understand through liver metabolism, for example, what kind of genotoxins are produced by diet, but there are many. And I think we need to be more comprehensive in understanding that. There's, for example, a recent observation by Jan Huymakers about how much life extension you get by calorie restriction. And it will be very interesting to see whether this actually contributes to avoidance of DNA lesions. There's some evidence for that, actually. So I think avoidance of at least environmental factors is very important. There are, of course, these inevitably occurring endogenous metabolic types. For example, formaldehyde is present physiologically in our organism, and it has distinct mechanisms of inducing DNA damage. And we need to learn better of scavenging some of that, probably. Scavenging the endogenous scavenging mechanisms. But we need to be much more mindful about what can we maximize the reduction of protecting us from environmental hazards.
Perfect, thanks. So we can avoid something. Shara, over to you. Do we have questions?
We actually do have quite a number of questions. So first, I would like to thank Prof Andrew and Prof Schumacher for that very exciting talk that you have. So to start off, we have one question. Actually, we do have quite a number of questions regarding epigenetic alterations versus genomic instability. So firstly, what exactly is the difference between the two in the hallmarks of aging? And what do you think is the relative contribution of DNA damages versus epigenetic alteration as a cause of aging?
Yes, so DNA damage can have epigenetic consequences. So, for example, we just recently showed that upon DNA repair, the epigenetic code is changed. And what happens is that the epigenetic modifications that occur after the repair event, for example, during transcription blocking lesions that are very important for aging, you see a specific deposition of a very specific histone modification that is now required to reinstate protein homeostasis, to reinstate the normal physiological state of cells and the entire organism. And this directly relates to the regulation of protein biosynthesis, protein homeostasis. So it actually, the epigenetic consequences are really a link then from DNA damage, its repair, to homeostatic failures in that occur in the aging organism, and that occur as a consequence of DNA damage accumulation. So the epigenetic mechanisms, chromatin remodeling factors, for example, are intricately involved in the DNA repair process. So they also affect the repair mechanisms during the repair reactions. There are remodeling activities. So it really has, and also the chromatin state that is epigenetically regulated, then also determines the sensitivity, the susceptibility to DNA damage, heterochromatin versus open chromatin. So there's a very, very tight interaction between DNA damage and epigenetics.
Thank you so much for that. I think I do agree as well that most of these are all really an interplay. All right, so the next question that we have, UV is sensed by the neurons in the worm and produce ROS. Since SKIN1, NRF2 is phosphorylated by PMK1 in MAPK and also responds to ROS, does SKIN1 play a role in this stress response?
So we looked at skin one in the context of the somatic response to DNA damage, and there we did not find an involvement of skin one. We haven't looked at that in the context of the germline DNA damage. But in the SOMA, we have actually extensively looked at different skin one mutants and things like that. But it would be interesting to further investigate that also with relate to the crosstalk between the SOMA and the germline. We haven't done that yet.
All right. We also have another question regarding reproductive organs. So the loss of reproductive organs appears to be beneficial for male longevity, whereas menopause in women brings a lot of issues. So do you think menopause actually increases longevity in women?
So there is a relationship between the age of menopause and lifespan in women. So the later menopause occurs, the longer the lifespan is. So there's at least this correlation. That, of course, doesn't mean that there's a causal relationship between that, because both could be associated with the longevity type. But I think that would at least argue that there's a relationship between reproductive aging and systemic aging, which is something I think it's very important to investigate. What are these communications? And it is very, very consistent with the observations in C.elegans that the germline determines somatic aging. So if the germline is compromised in its ability or requires more time for reproduction, which would be extended menopause, essentially, longer fertility period, that this would then promote the longevity of the soma. So of the in humans of the systemic, it would slow down systemic aging. I think these interactions are extraordinarily important to figure out. In mammals, unfortunately, they are much more complex than in worms, but I think we can lay the groundwork here to really understand this better.
Okay, thank you so much. All right, so another question that we have is this DNA damage response between somatic and germ cells a two-way mediation or communication?
Yes, absolutely, absolutely. And I showed you PMK1, this P38 MAP kinase in the intestine, but it directly crosstalks to another MAP kinase, ERK12 MAP kinase, in the germline. So there's a very tight, very complex type of interactions between these two distinct MAP kinase signaling pathway, one in the germline, one in the intestine. This clearly goes two ways. And the initiating signal comes, of course, from the germline, because the germline experiences a DNA damage. And we showed that, for example, with the meiotic recombination mutants, where we really only have extra DNA damage in the packaging cells in meiosis during oogenesis, and that is enough to trigger then the PMK1 response in the intestine. So it goes either way, indeed, yes.
Last question, Shara.
All right, okay. So I think this is one of the interesting questions that we have from David. How close do you think we are in finding realistic and practical therapeutic treatments in preclinical studies that can be effectively used to target DNA damage for healthy longevity?
We are on the way to get there. We have some very exciting concepts to look forward to. I think this will really start to become a new era of really fundamentally tackling the problem of DNA repair. And I'm optimistic that currently we're not there yet, but we are on a good way. And I'm optimistic that we will see that within this decade to really advance into the direction of the clinic.
Thank you so much, Bjoern. Also with this very enlightening closure of your talk, that there will be absolutely medication. And until now, I would say let's avoid the external stressors. And there was the DNA damage, of course. Thank you so much. It was very nice. And the C.elegans data you have are beautiful. Hopefully you also enjoyed the audience of this talk. Please use the chat function for your comments and feedback. We always look at your feedback and try really to implement it. If you also have an idea who we should invite, please let us know. At the end of the show, we will also show you some news from our center in the School of Medicine. Of course, the next session will be February 17th, that's next week. And Professor Brian Kennedy will be the host. And the next speaker is Professor Richard Seo from the King's College in UK. Every time we are closing with a video, of course, we are also doing that today. And this video will show you friendships and friendships of two generations, which are quite wonderful, I must say. So take care and enjoy friendships.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.